Hi, my name is Wuxian Shi. I'm a structural biologist, and I also work as a scientist at the National Synchrotron Light Source 2. Today, I'm going to talk about a study on blocking a key step in viral replication in SARS-CoV-2. SARS-CoV-2 is a virus that caused COVID-19. Since the beginning of COVID-19 pandemic in 2019, the disease has killed over 6.9 million people in the world. Now, there are effective vaccinations that protect people from getting seriously sick. However, even a fully vaccinated person can still get infected. So there is still a need to develop new treatments and new drugs against this disease. This is a life cycle of SARS-CoV-2. Now, it looks a little bit complicated, but it can be divided into three major steps. First, the virus binds to the receptors on the surface of human host cell and enters the host cell. Then, the virus will make its own proteins and then make these enclosures that allows the replication of itself. And after that, the virus repackages itself and exit the host cell in multiple copies. So today, I'm going to focus on the second step, where the virus replicates itself. So this particle you are seeing, or enclosure you are seeing, is absolutely required for the replication to occur. If we find a way to block the formation of these enclosures, then we find a way to block the viral replication. So this shows a simple cartoon of the formation of the DMB enclosure. It starts with the membrane from the host cell. The host cell membrane will double up in this membrane pairing step. Then this double membrane string will curl up in the curvature step, allows the fusion of the membrane to form this separate entities where the viral replication will occur. So how the virus does this? The virus will make its own protein, like the protein showing in orange color. The protein is embedded in the membrane and forming a locking interaction with another protein or directly with the membrane to draw the membrane close together to form this curvature. So now, I'm going to focus on a protein that is critical in this step called CoV-Y protein. We would like to study the structure of the CoV-Y protein, and based on that information to understand the function of the protein, and then to develop drugs to interrupt the CoV-Y function to stop the viral replication. So this is a work from Dr. Bing Hao's laboratory from University of Connecticut. They use this method called micromolecular crystallography to study the structure and function of the CoV-Y protein. So this is a flowchart of the method. You start with the protein crystal and shine X-ray on the protein crystal and record the diffraction from the protein atoms in the crystal. And with that diffraction pattern, we're able to resolve three-dimensional structure of the protein. So why protein crystal? Because in the protein crystal, all the protein molecules are arranged in an orderly manner. So the atoms from the protein, they diffract, the diffraction from the atom of the protein overlay on top of each other, so making the diffraction more intense, allow us to record the diffraction pattern. So you all have seen crystals of a salt or crystal of sugar, and those crystals are large enough for you to see them. They are also pretty tough. Protein crystals are very different. You likely will need a microscope to observe a protein crystal, and also protein crystal is rather fragile. So why is that? Because the protein, a protein molecule is thousands times larger than a salt or sugar molecules. So it's really difficult to arrange the protein molecules in orderly manner in a crystal. So it's hard to grow protein crystal. So now I'm going to show a collection of protein crystals. And these are beautiful crystals, and they have sharp edges, and they also have some symmetry to them. So these are type of the crystal you would like to have when you try to solve a protein structure. However, in most of the cases, these are the type of the crystal you're going to get when you try to grow a protein crystal. So like needles, showers of small crystals, or crystals that are so thin and then stacked together. So what you're going to do when you have crystal like that, then you come to the two crystallography, micromolecular crystallography beamlines at NSLS-II. So at this beamline, there are two important characteristics for to help user to be able to work on difficult crystals, like the crystals that you're seeing here, like very thin needle clusters and plate that are so thin, it's about 1 micron in thickness. So we are able to focus the beam down to 1 micron, and that allows people to work with crystal in 1 micron size. We also have high level of automation. The highest throughput is about 25 samples per hour. On the right, you're seeing a automated data collection recorded at AMX. So we offer a fully automated data collection. Like at 10 o'clock in the night, you can set up a fully automated data collection. And by 8 o'clock in the morning, you could get up to 200 data sets collected. So this is particularly useful when you have a project that requires you to screen hundreds or even thousands of crystals. So that's about the method. Now we're coming back to the COVE-Y structure. The Hall Lab solved the high-resolution structure of COVE-Y. Now you're seeing the structure in space failure model. From the side at the left, the protein is called COVE-Y protein. It actually has a Y shape from the side view. The functional important pieces of the protein are colored in red. So if you rotate the structure down by 90 degrees, you'll see the top of the protein, basically the top of the two arms of the protein. Now you see clusters of the red pieces there. So this indicates that the functional site of the protein is on the top of the two arms of the Y-shaped protein. Now remember that, and the Hall Lab take a step forward. They screen a large libraries of small molecule compounds and try to find the compounds that can fit into cavities of the protein. So here shows two sides. Let's focus on the side one that is right between the two arms of this Y-shaped protein. If you remember from the last slide, the functional site of this protein is on the top of the two arms of the Y-shaped protein. So now if you have a compound that wedge between the two arms, that potentially can change the angles of the two arms, and then that change the shape of the functional site. So what is the implication of this? Now we go back to the enclosure formation that allows the viral replication. So the COVE-Y protein is this in the light orange color. It forms a locking interaction with another protein or directly with membrane. And that allows the viral replication to happen. So now if we change the functional site or shape of change the shape of the functional site of COVE-Y protein, now it won't be able to form a tight interaction with the other protein. And now the double membrane string will stay in its stringy conformation. And that will not, the viral replication will not occur in this case. So to conclude, the high-resolution structure of COV-Y protein has been determined. Using that information, potential drugs have also been identified to stop the viral replication in SARS-CoV-2. For the future direction is basically to refine the compound to bind the protein more tightly or more specifically, and then further develop these compounds into potent drugs against SARS-CoV-2. Here's a acknowledgement. The work is mostly done by Dr. Bean Hall's lab from University of Connecticut. The staff from NSLS-II contributed collaboratively to help to solve the structure. The beamlines were operated by CBMS team that's supported by NIH and BER.